So yes, good evening, everyone. This is Dr. Abhijeet, Faculty of Biochemistry at Oracle. So we have initiated Oracle 21 days revision series live class for university exams. So the topic that I will be discussing today, it is the biological membrane and transport. And this topic I will be covering like entirely the most important and the potent questions that have been asked in the previous 10 years, right? I will be discussing the important topics in the form of question and answer because anyways, at the end, guys, you have to just focus on answer writing. So I will be writing the entire answer. I will be discussing the entire answer, right? I will be discussing the topic via like I'll be explaining the concept, right? So it is beneficial for the ones who are going to appear for the university prof exams, right, which are going to take place soon, and the ones who are going to give the INI exam, or they are going to give any like FMG exam or NEET PG exam, they can also benefit from this particular session, right? So yeah. So the topics that I will be discussing today in the form of questions, it is the biological membrane, the plasma membrane. Obviously, it is a very, very important question that has been asked in the prof exams multiple number of times in various university throughout India, right? So yeah, long essay question, it will be in the form of a 10 marks question. Then I will be discussing 3 marks. Okay, short essays, maybe like 8 marks or 5 marks question. I will be discussing the membrane protein, the transmembrane protein and everything, right? I will be discussing the process of osmosis and the mechanism of ion channels and their regulation. Apart from that, I will be focusing on very short answers that are like basically 2 to 3 marks question about aquaporins, like what is their role in cellular function, the membrane potential and the physiological importance. All the membrane potential, it's a part of physiology more, right? And I will be discussing the concept of membrane fluidity, which I will be some part I will be covering in the very first question itself, right? Now, guys, as you all know about the Oracle app, for flexible learning, guys, we have these recorded versions. See, we are conducting live classes daily from 6 p.m. to 10 p.m. live on YouTube, live on the app. But in case if you miss the live classes, you can just go on for the recorded versions, which are available on the app. The live classes we are conducting daily from 6 p.m. to 10 p.m., right? Different subjects, different educators, right, on a single platform. Apart from that, the practical sessions, many of the students inquire what to do just before the practicals. So we have the recorded versions of the practical sessions along with the practical notes. So you can just go on for these recorded versions which are available on the app. Next is the exam module. 1 month before the exams, the students are in a very anxiety and panic state, know what to do. So, sir, we cannot read the book. It's too like not enough time to go through the book. So we have no like cover the entire biochemistry, all the important, potent questions which I feel they are important for the upcoming exam are being covered under 20 hours, right? So that is you can go on for this express exam module. And the last one, that is the most important one, that is the hard copy book set, which you can order from your home. It is like way easy and it is available at your doorstep. These hard copy notes contains all the like topics which are there, right? In the form of question and answer. We have fun zones, we have riddles, we have MCQs covered over here. We have taught you the way how to approach to the question, whether it is a long question or a short question, right? So these notes will be an additional aid to your success, guys, right? Coming on to the next one, that is the foundational classes, guys. You can always go on for the foundational classes for the flexible learning, right? Apart from that, I have already told you regarding the live classes. It is from 6 p.m. to 10 p.m. daily, right? We are covering the, yeah, different subjects on a single platform. So you can always go on for that. We have the express exam module under 20 hours. Then we have one-to-one doubt clearing session. If you guys have any doubts related to any particular subject, right? Any particular question, any particular topic, you can always book a prior appointment with that particular faculty and you can get your doubts clear then in that, right? So we have the hard copy book set that is being available at your doorstep. You can always go on to the website and know on the app, you can just book on for these notes, which will be an additional aid to your preparation. We have the question bank, guys, from basic to advanced level. Obviously, after each and every topic, you will be getting certain questions, right? And these questions are being curated by the expert team. Apparently, guys, nowadays the clinical vignettes are in demand. So we, the educators over here, we have created those clinical vignettes also, so that you can just go on for the practice. Even the 10 years, past 10 years question papers are being covered from like different universities we have collected. So we have reduced the burden of yours also, to go and search on for those examination papers. We have already curated like those MCQs are being included in this part as well. Now, practical sessions, I've already told you it's being recorded. The recorded version is being available on the app. Now, the subscription plan, we have the foundation plan, guys. We have the advanced learning plan. We have the complete learning plan, and we have the ultimate MBBS plan. You can use my code Dr. Abhijeet20 for additional discounts. Now, coming on to the very first question, guys. Analyze the structure and function of the biological membrane, including the fluid mosaic model. Now, guys, you already know every cell is basically covered by a membrane that is called as the cell membrane. Okay. So this cell membrane, it is called as the plasma membrane. This cell membrane, let us suppose that this is the cell, and the cell is basically covered by the cell membrane. It is called as the plasma membrane, or it is also called as the biological membrane. It is also called as the biological membrane. Now, the contents of the interior of the cell, they are basically separated from the outer environment, okay, by the help of this particular membrane only, right? Now, they regulate the movement of molecules, guys, right? Now, let us just try to understand what is the structure of the cell membrane. And guys, always try to remember structure of this biological membrane. So basically they are covered, okay, or they are composed of majorly three things. We have the lipids, proteins, and carbohydrates. Carbohydrates. Now coming on to the lipids guys, the most common lipid, okay, most abundant lipid in the cell membrane, it is nothing but the phospholipid. Phospholipid. Now, this phospholipid, it is basically not existing in a single layer. It is existing in a double layer. So we always say phospholipid bilayer. Okay, phospholipid bilayer. Basically, now guys, over here, you just have to remember the structure of the phospholipid also. But always, in any answer writing, guys, I always ask students to draw a schematic diagram. It's very important, guys. I'm not asking you to draw something very fancy. Yeah, but a schematic, correct labeled diagram would help you to fetch more marks, okay, than just writing paragraphs with no sense, right? Now, if we talk about the structure of the phospholipids, better remember that the structure of the phospholipid, it is having one head and we have two tails, guys, over here. We have two tails and one head. Two tails and one head. Now, remember guys, the head part, it is polar. It is hydrophilic. Hydrophilic. Okay. Now, the head part, it is water loving. So it is arranged in such a way, guys, that the head part always faces towards the aqueous compartment or the aqueous or the water side. They are arranged like this, the tails facing inside and the head is always outside. So you already know, if let us suppose that this is the cell membrane, right? If this is the cell membrane, yes. So it is like this. Now you can always see the head part. Internally, the cell will also have some fluid that is called as intracellular fluid. Intra means inside the cell, and the cell outside the cell, we will also have some fluid that will be called as the extracellular fluid. Extracellular fluid. So just fluid means the water part. So always the head, if I'm saying it is hydrophilic, philic means the water loving part, the charged part, it will be always situated outside, right? And also the second layer, the head will be situated towards the intracellular fluid, right? Keeping the tails inside, right? So remember the tails are hydrophobic and non-polar. Now, this arrangement is very, very important for the selective permeability of the membrane, right? And yes, to maintain a stable environment internally, it is like very, very important. We also have proteins, guys. We also have proteins, but just on what like in the notes what they have mentioned, we'll just try to focus on these points as well. Remember that the plasma membrane, it's a protective sheath, it's an elastic structure, it envelops the cell body, right? It separates the extracellular fluid, the ECF, from the intracellular fluid, right? Now, there are various lipids like phospholipids, sphingolipids, and cholesterol. Remember, beta phospholipids, it is the most common lipid found in the cell membrane. And when I say most common, it is the most abundant lipid also. Most abundant lipid also. Now, when I say sphingolipid, beta, over here, the sphingo, what basically means the sphingosine. Sphingosine is nothing but the alcohol. Usually the alcohol, okay, you already know we have the free fatty acids and glycerol, the alcohol, right? The free fatty acids and the alcohol. This alcohol either can be glycerol or it can be sphingosine. So yes, we have sphingolipids also. Okay, it's a compound lipid. Compound means the complex lipids that we have discussed yesterday. We have cholesterol also, right? So basically the lipid component not only includes the phospholipids, we have the sphingolipids also and the cholesterol also. Coming on to the next one, that is the membrane proteins. Now, membrane proteins are embedded or associated with the lipid bilayer. Within the lipid bilayer, we have the integral and the peripheral proteins. Okay. Now, when I say integral protein, guys, remember they span the entire membrane. They are present all over the membrane. For example, let us suppose this is the lipid bilayer, right? This is the lipid bilayer, and these membrane proteins are present like this. So they are covering the entire, like this plasma membrane. So these are nothing but the integral proteins, right? Now these integral proteins, they can either function as ions, I will be discussing that, or they can either function as channels. They can either function as ions or they can either function as channels also, right? Now, these proteins basically allow the transport of some molecules from ECF to ICF or from ICF to ECF, right? And when we say some molecules, it means the selective permeability of the membrane. It will only allow some molecules to pass from outside to inside and vice versa, right? Coming on to the peripheral proteins. Now remember that these peripheral, obviously the name suggests periphery. They are either located outside or they are located inside. Okay, they may be present outside or they may be present inside. Okay, so these are nothing. They are just located on the periphery. They are called as the peripheral protein. Now these peripheral proteins, they actually help in cell signaling, even they help in maintaining the cell shape. They help in maintaining the cell shape. Coming on to the carbohydrates, guys. So remember that they are present on the outer side, unlike the peripheral proteins that are present on both the sides, right? Either on like the outer part and the inner part. The carbohydrate is present only on the outer part, guys, right? And carbohydrates are nothing but these glycolipids or glycoproteins, okay? And they together basically form the glycocalyx. Glycocalyx. Now, the carbohydrates helps in the cell recognition, right? The cell signaling. They also helps in protection because they form a protective layer. For example, if you can just see on the membrane over here, we have the glycoprotein. Okay, so you can always like go on for this. The carbohydrate sugar chain, they are just present on the outer membrane, giving up the protection, right? They help in signaling. They act as a signal molecule, right? So that is like you have to remember. Now coming on to the fluid mosaic model, according to the knowledge, guys, you already know from +1 that they are being proposed by the Singer and Nicholson in 1972, right? So they describe the structure of the biological membrane. Now, comparatively, beta looked at the biological membranes are very, very thin. They are very, very thin. They are ranging from only 5 to 8 nanometers. They are ranging from 5 to 8 nanometers in thickness, right? So they are composed of lipid bilayer. We have already told you, right? And we have the double layer of lipids, right? Where the head of the phospholipid faces towards the aqueous environment, whether it is inside or outside. Now, so remember the phobic means water heating part will be basically facing the entire or the outer water like fluid, right? Outer water fluid. Now, so that is something we already know, the hydrophobic and the hydrophilic part we have already discussed. Just I wanted to make more regarding these proteins, guys. These proteins, they are actually very, very important. So remember the example of extrinsic protein, again, a very important MCQ question. It is cytochrome C that is found in which organelle of the cell? That is the mitochondria. Right? Intrinsic proteins, basically, they are tightly integrated to the lipid bilayer, right? So remember the integral proteins are nothing but these, you already know, are the hormones, the receptors of the hormones, right? And cytochrome P450, right? So you will be also studying this and know the when you will be studying the drug in the pharmacology in the third year. I mean, in the second year, you guys will be dealing with the cytochrome P450 and all these hormonal receptors and everything again. So yes, remember guys, the lipid bilayer, it is basically no exhibiting a asymmetrical nature. Okay, it is exhibiting a asymmetrical nature. Okay, because the proteins are not evenly distributed. Some proteins are like, for example, integral proteins are no placed just spanning throughout the membrane. Some are located outside, some are located inside. So this gives a asymmetrical nature of this biological membrane, right? So it gives like a mosaic like appearance, right? That's why it is called as the fluid mosaic model, right? So basically mosaic means the proteins and the fluid basically the lipid part, right? So this is like you have to remember, guys, right? Now you can obviously see the peripheral protein, right? You can see the integral protein over here, the transport protein, okay, the peripheral protein, right? Now that was regarding the first question. Again, in this question, guys, 2 3 points that I wanted to mention: the function of the biological membrane, right? That has not been mentioned. Just in short, guys, what you have to mention: function of the biological membrane. The very first one, it is the selective permeability. Now, the membrane will act as a selective barrier, guys. It will only allow some molecules to go from outside to inside, right? And from inside to outside, including the ions, the gases, right? The nutrients, the drugs, and the waste products. Now, guys, you already know when I say selective permeability, the non-polar, when I say non-polar, the uncharged, the non-polar small molecules. Now, when I say small molecules like oxygen, carbon dioxide, they are very much soluble in lipid. So they can easily cross the membrane, right? For example, if we talk about the polar molecules such as the glucose, okay, glucose is very, very large, right? So it cannot cross the lipids easily. So this glucose basically require a transport protein. And you already know from your +1 knowledge that this transport protein is nothing but GLUT, that is the glucose transporter. We have various types: GLUT1, GLUT2, GLUT3, GLUT4, GLUT5, 7, right? So that is something you have to remember. Even now, this transportation of membranes is also one of the important function of this. This is basically controlled via two processes, guys. That is passive and active transport. I will be covering these parts in the upcoming question. Passive and active. You just write the term ATP and your answer will be done. Where ATP is required, it is called active transport. Where ATP is not required, it is called as passive transport, right? So that is something important. Even one more function, that is, yes, the membrane also allow the internalization of some compounds, right, through endocytosis and exocytosis. Endocytosis and exocytosis. Now, these processes are very no important for waste removal, right? These processes are very important for waste removal. Okay, these are important for no uptake of the nutrients, right? And they are important for communication between the cells, right? So remember, guys, yeah, the biological membrane also helps in maintaining the homeostasis of the body, maintaining homeostasis. Now, by regulating the flow of materials from inside to outside and to outside from to inside, guys, vice versa, they are maintaining the homeostasis, guys. No, basically, so that the internal conditions of the cell are basically stable. Okay, they are remnant. Now, that was regarding the very first question, the structure and the function of the biological membrane, including the fluid mosaic monitor, right? So, yes, coming on to the next one, that is, yes, discuss the role of the membrane proteins in facilitated diffusion and active transport. Now, guys, we already know we have two types of transport, right? The active transport and the passive transport, right? You already know from your previous knowledge, we have two types of transport, the passive and active transport. One of the type, when I say passive transport, guys, you already know the ATP is not required. Very important. And the active transport, the ATP is required. One of the examples of passive transport, it is the facilitated diffusion. Facilitated diffusion. Facilitated diffusion, right? No. 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 Facilitated diffusion basically allows the movement of substances from high to low concentration. Movement of substances from high to low concentration without the involvement of any ATP molecules, without the involvement of any ATP molecule. Okay. Now, facilitated diffusion cannot take place without any protein, guys. And these proteins are called as channel proteins. Very important, right? Channel proteins. They require the help of channel proteins or carrier proteins okay, to diffuse into the lipid bilayer. For example, you already know we have large substances, we have small substances. Okay, not small, actually, we have the polar one. Now, guys, if we say about phospholipids, lipids are not soluble in water, right? So lipid will never allow water to come inside the cell. So we need those protein molecules to know for the transportation of some charged substances which are having charge, right? Which are large in size, which cannot easily diffuse into the lipid, right? So these proteins are very, very important over here, right? Now, if we talk about channel proteins, guys, in short, if we talk about the channel proteins, now these channel proteins forms the aqueous pores. Okay, they will create holes, they will create water holes, right? These are called as channels, right? Or channels. These now they span the lipid bilayer. They are present all over the lipid bilayer. Guys, remember they allow specific ions. Now, when I say specific ions or small molecules to pass through them. Now, specifically, if we are talking about the ion channels. For example, you already know the sodium ions, the potassium ions, the calcium, the chlorine. So these ion channels can be gated, right? They can close. Now, when I say gated, guys, it clearly means that they can open or close in response to stimulus or in response to any signal. Okay, like for example, there's a change in the voltage, so they are called as voltage gated channels, right? So if any no substance is bound to the ligand, so they are also called as ligand gated channels, right? So that is something you have to remember. One more channel protein that you can remember over here, it is the aquaporins, right? So this aquaporins, basically aqua means water. So it will only allow the water molecules to pass through them, and that too in a very rapid fashion, that too very fastly, guys. Right? So that is the channel proteins, right? Now we have carrier proteins also. Now, carrier proteins are the ones that go very conformational change, okay, in response to any stimulus or no, if they bind to any particular molecule, right? So if they undergo a conformational change, they will always allow that molecule to no go inside the cell. For example, most common example is the glucose transporter, right? The glucose transporters. Okay, so these glucose transporters basically they help in the transport of glucose into the cell. Okay, responding in response to the concentration gradient, guys, right? For example, in blood the glucose levels are very high, and in the cell the glucose levels are low. So facilitated diffusion typically means that the glucose will be now flowing from the high level or the high concentration to the low concentration, guys, right? So yes. Now, what are the characteristic of these facilitated diffusion is like they will only allow certain molecules to pass through them. They will not allow each and every molecule to no cross it. And secondly, there is no ATP requirement over here, no ATP requirement over here. And guys, there is something called as saturation. Now, if the glucose levels are no obtained by the cell in a very sufficient manner, it will the concentration gradients are almost the same, then this facilitated diffusion will automatically stop, right? So that is something you have to remember about the facilitated diffusion. Coming on to, yes, I just wanted to discuss over here. These are all given in the notes also, right? So yeah, the same things that I have discussed over here. Yes. Hmm. Now coming on to the active transport, guys. So you already know the substances are moving from low concentration to high concentration, and it is that's why it is called as the uphill transport from low concentration to high concentration. And basically in active transport there is always requirement of this ATP, right? Yes, it requires energy, and energy is basically no, it is taken up by the breaking down of this ATP, that is the adenosine triphosphate, right? So the carrier proteins which are involved, they are of two types in active transport. We already know uniport or symport or antiport, guys. Now, uniport is a carrier protein which will only transfer the molecule in a very single direction, either from outside to inside or from yeah, or symport or antiport, basically what it will do, yes, the symport or antiport, one molecule will be no going from inside to outside. Similarly, the other molecule from outside will come inside, right? So it is a carrier protein that can transport two substances at a time, right? In a very antiport means in a very no opposite direction. Anti means opposite, or symport means when both the substances from outside are going together inside. So that is called a symport. Synergistic action. Basically, they are combining the action and they're just no moving like this. So active transport, again, it is basically of two types, the primary and the secondary transport. The primary and the secondary transport. Primary transport is where the energy is liberated directly from the breakdown of ATP. You already know the sodium, potassium, calcium, and hydrogen, the chloride, they are transported by these method, and the pump is known as sodium potassium pump. The sodium is known as the pump is known as sodium potassium pump. Now, what is this pump? Basically, the sodium potassium pump is one of the most well known examples of active transport. It no, it moves the three sodium out of the cell, right? From your physiology knowledge, and two potassium inside into the cell, and that too against the concentration gradient. Against the concentration gradient. Against the concentration gradient, right? So yes, by the breakdown of hydrolysis, by the hydrolysis of the ATP, basically this energy is required just to move three sodium out of the cell and two potassium inside the cell, right? So yes, the another method of the active transport, it is the secondary transport. So basically, it is transport of a substance with sodium ions by means of a carrier protein, right? And remember, guys, it may be in the same direction of the sodium movement or in the opposite direction, right? So it can be a co-transport or it can be a counter transport, right? Co-transport or counter transport, right? So that is you can just see, for example, the most common one, the sodium, potassium, and chlorine. So they are moving again in the same direction, sodium, potassium, chlorine co-transporter, right? Potassium and chlorine moving together out of the cell. It is potassium chlorine co-transporter, right? Sodium and neurotransmitter co-transmitter. For example, neurotransmitters like GABA or dopamine, basically they're moving together inside the cell. It is called as sodium neurotransmitter co-transporter. But if I see that in opposite direction, for example, sodium moving inside the cell and potassium or no, or calcium moving outside the cell, it is called as sodium calcium exchanger. Or for example, sodium moving inside the cell or hydrogen moving outside the cell, it is called as sodium hydrogen exchanger. So either it can be in the same direction or either it can be in the opposite direction also, right? So that was regarding the second question. Now coming on to the third one, that is the process of osmosis. I think from +1 you guys are studying this. It is the passive movement. Now guys, when I say passive, you already know that there is no requirement of energy. Remember, it is the passive movement of water molecules, okay, across selectively permeable membrane. Selective permeable membrane. Okay, that is the cell membrane. Okay, it is from, yes, from area of low solute concentration to an area of higher solute concentration, right? So such that the equilibrium is reached, right? So yes, that is something you have to remember, right? Now, guys, there are two things that you have to understand over here. Now, what is the process of osmosis, guys? The very first thing you have to remember that there is something called a selective permeability. Now, the membrane is selectively permeable. It allows some molecules like, for example, water to pass easily, right? To pass easily while restricting the movement of other molecules like large solute molecules or ions. Now, when I say water, guys, now water basically will transport from a region of lower concentration of solutes. Again, this makes sense, guys. Not from higher concentration, it is lower concentration of solutes. Now, जहाँ पे solute कम होगा, obviously there will be higher concentration of water. there will be higher concentration of water, right? Higher concentration of water. And where there will be higher concentration of solutes, obviously, if we take a very common example of salt and water, if salt is less, obviously the water will be more. And if salt is more, then the water will be less. So in just this manner, you can remember that the lower concentration of solutes, we have the higher concentration of water, and automatically we have the higher concentration of solutes, we have less concentration of water, right? Now, this movement basically tends to stabilize both the solute concentrations as well on both the sides, right? The typical random movement of the water. And apart from that, remember the osmotic pressure. Osmotic pressure. Now, osmotic pressure, we always tend in in biochemistry, it is the extra pressure that is applied to stop the process of osmosis. For example, even the water, aqua water, the filters, the water filters, they are actually working on the process of this reverse osmosis, right? So basically, this osmotic pressure, it is directly proportional to the concentration of solutes, right? So yes, that is something you have to remember. Now, now, guys, if we talk about the real life example of osmosis, one is the very common one, that is the kidneys, right? The kidney function. Now, osmosis is something very, very no important for the kidneys because it helps to maintain the water balance, right? By either by concentrating the urine or by diluting the urine, right? So the kidneys are basically using the process of selective permeability, right? And so that the water can be reabsorbed back into the body, or the excess water that can be easily no removed by the body, right? So yeah, these points you have to include in your answers. No, yeah. Coming on to the next one, that is, yes, the mechanisms of ion channels and their regulations, right? So yes. Now, when I say ion channels, guys, when I say ion channels, so remember that ion channels basically they form or they function as tunnels. They are just like tunnels, okay? And these tunnels within the cell membrane. Now, let us suppose that this is a cell membrane, and these are nothing but the tunnels, right? So remember, now, first, how they are regulated, guys, they worked on something called as selectivity of the ion. Selectivity of the ion. Now these channels which are working, or these channels which are working as tunnels, they are actually specific for a specific ion. For example, if I say sodium channel, so it only it selectively allows sodium to pass, sodium ions to pass. Calcium channel, it selectively allows calcium to pass, for example, chloride, or for example, potassium, right? So yes. That is first the selectivity that you have to remember, that a particular channel will only allow a particular ion to pass through that channel. Secondly, guys, you have to remember one more important thing over here, that is called as the gating mechanism. Now this gating mechanism, it is basically of two types, guys, right? The voltage gated ion channels, and the ligand gated ion channels. Now, when I say voltage gated in physiology, guys, you already know they open or close in response to the changes in the membrane potential, right? For example, If I commonly take the sodium, okay, the sodium channels or the voltage gated sodium channels, they only see, if we talk about the resting state, the sodium channels are closed. The sodium channels are closed, right? Now, if there is a action potential, action potential, now what will happen, right? Now, action potential, obviously, when the membrane will depolarize, there will be an action potential. This channel, this voltage gated sodium channel will undergo, yeah, conformational change, okay, and it will open, right? It will allow the sodium to flow in the cell, right? Comparatively, if we talk about the ligand gated ion channels, guys, remember that they open or close in response to binding of a specific molecule, right? For example, ligand basically means the neurotransmitter. For example, it can be a neurotransmitter. Now, over here, if I have to take any example, yes, the one which I can remember, it is nicotinic acetylcholine receptor. Now, this nicotinic acetylcholine receptor, it is basically a ligand-gated ion channel. It will only open, guys. It will only open when no acetylcholine will come and bind to it. When acetylcholine will come and bind to it, it will open. Then it will allow the movement of sodium from outside to inside, initiating a response. Right? So that is something you have to remember. Right? Now coming on to aquaporin, guys, you already know about the aquaporin. It is a integral membrane protein. Aquaporin, we have already discussed this. Yes, it is an integral membrane protein. So it forms a channel, guys. It forms a channel, and that is for the transport of water. That is for the transport of water molecules across cell membrane. Yes, across cell membranes, right? So, yes, if we talk about the structure, it is basically composed of 6 transmembrane alpha helices, right? So it forms a pore channel, right, in which water no molecules can pass, right? So we have various no aquaporins. Now, depending on the function, guys, depending on the function, we have various types of aquaporin. For example, aquaporin 1. It is found in red blood cells, right? It is found in red blood cells. It is found in the kidney tubules. Now, when I say kidney tubules, I'm talking about the nephron tubules, the proximal convoluted tubule or the distal convoluted tubules, right? And in certain epithelial cells also. So basically, if they're found in kidney tubules, what will be the function, guys? What will be the function? It will help in the reabsorption of water. It will help in the function of reabsorption of water. Now, the another one, aquaporin 2. Aquaporin 2, basically it is found primarily in the kidneys, and especially which part of the kidney? It is the collecting duct. The nephron, it is also having the collecting duct. So this collecting duct, it is basically regulated by a hormone that is very important. That is the, yes, ADH hormone, antidiuretic hormone, which helps in the reabsorption of water. Okay, so remember what is the function of ADH? It is helps in the reabsorption of water reabsorption. Now coming on to the third type, that is aquaporin 4, guys. It is found in the brain. And which cell of the brain? That is again a very important MCQ, that is astrocytes. Astrocytes, right? So it basically helps in regulating the flow of water in the central nervous system, right? We also have aquaporin 3 or aquaporin 5. They are basically found in epithelial tissues like skin. Skin, it is the largest organ of the body. They are also found in salivary glands. They're also found in lungs, right? They actually help to maintain the water balance within the cells. It's a short type of answer. You can just give the classification, where they are present and what function they are performing. Membrane potential, guys, you can just go on for the physiology one. That is like the same membrane potential. Moreover, they won't ask you this particular question in the biochemistry. One time they have asked, like, then we have just included this. So that you can just go on for that. Membrane fluidity, guys, I have already explained that. Yeah, membrane fluidity, we have already explained that. I've already like gone through it. So yes. So the factors that influence it, guys, that is something which you have to remember. One is the lipid composition, right? So yes, the length of the fatty acid chain, guys, basically when we say in phospholipids, right? It actually no influence the membrane fluidity. For example, we have short chain fatty acids. Now, when I say short chain fatty acids, basically I'm talking about the carbon number from let us say like 14 to 16, right? So they result in increased fluidity. Okay, why? Okay, they have increased fluidity because shorter chains have less interaction between them, allowing them to move freely. On contrast, if we have a longer chain, okay, or we have something called as the longer chain fatty acids, for example, like carbon from 18 or 20, right, or more than that, they have more surface area for interactions, right? They have more surface area for interactions, and that is something they are leading to decreased fluidity, right? Even the second one that you can remember, the cholesterol, guys. Cholesterol, it is basically a type of lipid that helps in modulating the membrane fluidity. Okay. So yes, it is acting like a fluidity buffer. If there is a slight change in the buffer of the fluidity, it will help to try to maintain that. If we have high temperature, guys, so it stabilizes the membrane no by reducing fluidity, right? By reducing fluidity, right? So yes, and at low temperature, it tries to maintain it by, yes, increased fluidity, right? Now, even the membrane proteins, they also play a very important role in the fluidity, right? So we have already discussed the membrane proteins, the transmembrane protein, the integral, the peripheral proteins and all, right? So that is, you can just remember. So these are the main important ones which you can include. Obviously, it's a 2 mark question, so just write down about the lipid composition, the short and long chain, and the cholesterol part. That will be like sufficient. So that was regarding today's session. These are the various social media handles which you can follow and go on for recorded questions. Thank you.